Okay, hello everyone, and thank you for inviting me this year again to talk to you at the LabRoots Neuroscience virtual event. My name is Gemma, and I will be talking to you about patient-specific brain organoids for in vitro modeling of neurodegenerative diseases. So first of all, I would like to introduce myself a bit more. I come from Spain, from Barcelona, where I did my bachelor and master's in biomedical sciences. And then I moved to Germany for my PhD on stem cell biology, neuroscience, and neurodevelopment. This was my first contact with the field of stem cells and 3D cultures, as well as disease modeling using all these new technologies. And after that, I did a postdoc at the University of Luxembourg, involving also organoids and organ-on-chip models to study specifically Parkinson's disease, which I will talk to you about today. And since a year and a half, I'm a lead project scientist at OrganoTherapeutics. And OrganoTherapeutics is the spin-off company of the University of Luxembourg. And what we do basically is use our expertise on advanced three-dimensional stem cell models to test compounds against neurodegenerative disorders such as Parkinson's disease. So the learning objectives for today's talk are basically explaining about Parkinson's disease and its main pathological features. I will introduce the concept of organoids and assembloids, and I will explain how do we apply them to the study of complex neurodegenerative diseases. And last but not least, I will give you a couple of examples about how we use these models at the company to test compounds as potential treatments, especially for Parkinson's disease. So what is Parkinson's disease? Parkinson's disease is actually the second most common neurodegenerative disorder in the whole world, after Alzheimer's disease. And it affects about 10 million patients. And even though a lot of efforts have been placed into developing therapies, all the treatments currently available are only directed to managing the symptoms, and there is no cure available. Therefore, a big pillar in the disease care is the patient care, actually, which contributes to a big economic burden, which is estimated, for example, to $52 billion per year in the US, according to the Michael J. Fox Foundation from 2019. So probably by now is a bit higher than that. So consequently, there is a lot of interest in developing new and better therapeutic approaches for these patients. The most characteristic sign of Parkinson's disease, which is probably the one that you mostly known, is the presence of motor symptoms, which include tremors and postural instability. But patients also present a variety of non-motor symptoms, including sleep disturbances, loss of smell, which can actually appear long time before motor symptoms appear. The cause of these symptoms is the loss of a specific neuronal population, which are called the dopaminergic neurons, in very specific regions of the brain, more specifically in the substantia nigra, whose name is attributed to the presence of the dark pigment, which is called neuromelanin, and that you can see here in these in these sections of the brain, in the right-hand side panels, which compares the presence of these neurons, pigmented neurons, in a healthy person versus Parkinson's disease case, where the neurons are lost, as you can see by the absence of pigment. And the problem is also that the neurons are connected, not only in this brain region, but also connected to other regions of the brain, such as the striatum. And the loss of these connections also contributes to the pathological symptoms of the disease. And Parkinson's disease is a multifactorial disease, which means that there are different factors that contribute to the development of the disease. And only 5 to 10% of the cases have a clear genetic cause. There are several genes that drive the manifestation of the disease or that can increase the risk. But however, 90% of the cases appear sporadically. And age is actually one of the biggest risk factors for Parkinson's disease, which goes along with the increase in life expectancy for humans. So among other contributing factors are the environmental factors, as the incidence of Parkinson's disease is more frequent, for example, in industrialized countries, but also lifestyle is a big contributor, with diabetes, the diet, or sedentariness, for example, being shown as factors to contribute to the disease. So there are some factors that we can influence to prevent the disease. And as I mentioned earlier, most patients at the time of diagnosis already present motor symptoms. And this happens already when 40 to 60% of the neurons in the region I showed you earlier are already lost. And the disease can start up to 20 years earlier, as you can see in this graph, with non-motor symptoms that sometimes are a bit and are difficult to uncover. So, and what we are interested in is to actually going even further back and asking the question of what are the earliest changes we can find that can lead to Parkinson's disease. In that sense, we believe that there are defects that can already appear in a neurodevelopmental stage that are probably associated with specific mutations first as a first hit, which contribute to developing Parkinson's disease later on in life. However, there is a need for a second hit appearing later in life and probably correlated with all the other factors I mentioned earlier that can actually trigger the disease or not. And to study this hypothesis, we actually need models that are human-based and that are able to model or simulate these very early stages of life. The classical models that are used also very much or still to study Parkinson's disease are animal models. And they have improved greatly our understanding of the disease and the mechanisms that are involved in the pathology. However, these models do not always recapitulate the pathological hallmarks of the disease or its progression in humans. And the use of these models in the labs to study diseases are posing more and more ethical concerns. Another model that is available to study Parkinson's disease is adult tissue of patients affected by the disease. However, it is not hard. It is hard actually to access brain samples to study the disease, and these samples are actually post-mortem, which means yes, we can look at the actual tissue affected by the disease, but if you recall it correctly, this will be patients in which the neurons in these specific areas of the brain are already lost. So we are only looking at the end stages of the disease. And sometimes the quality of these samples is not the best due to preservation issues. So the problem is that a complex disease such as Parkinson's disease with multiple factors being involved cannot be studied just by looking at one gene or a molecule. And as I just said, some models don't recapitulate sufficiently what the human disease looks like. And there are no individuals, there are no two individuals that are the same, meaning that we need actually specific models to every patient, ideally. And therefore, there is the need to develop more complex human models to study the disease at these earlier stages. To facilitate this, about 10 years ago, Yamanaka and colleagues developed the technology by which fibroblasts, which are skin cells, can be from humans can be reprogrammed to a state of induced pluripotent stem cells, from which we can derive again most cell types of the body, allowing us to study the specific cells involved in the disease, such as the dopaminergic neurons, and allowing this study to be specific to every donor of these cells, be it healthy or a patient affected by Parkinson's disease or another neurodegenerative disorder. So we have a tool that allows us, in the case of Parkinson's disease specifically, to compare the behavior and the properties of dopaminergic neurons coming from an individual affected by the disease and a healthy one in the dish. And even though we can study like that easily the main neuronal population affected, the dopaminergic neurons are not alone inside the brain, and there are other multiple cell types which compose the brain and that can contribute to the overall pathology. And therefore, three-dimensional culture models have been developed to increase the complexity of the tissues that we have in the cell culture in an effort to bring them closer to the actual body physiology. And in this regard, organoids appeared as newer and more complex models to study diseases. What are organoids? Organoids are three-dimensional, self-assembled cellular structures which are generated from stem cells, which mimic the structure and functions of the organs they model. And with the years, there are different kinds of organoids that have been developed for different organs of the human body, for example, the brain, the lung, the intestine. And within the brain organoids, there are also a lot of efforts to develop region-specific organoids to study not the brain as a whole, but really specific regions affected by the disease. And in this regard, we actually focus on midbrain organoids, which are the organoids that model the region I was telling you about, which is the substantia nigra of the midbrain, which is the one affected in the case of Parkinson's disease. And in this case, what we do is we start with patient and healthy cell samples that were reprogrammed in this pluripotent stem cell stage. And then we further differentiate those into an intermediate neural precursor cell stage, which are still stem cells, but already committed to differentiate on the dish to the brain-like structures. And these are the cells that we use as the starting population for generating these organoids. And how this works is basically we place a certain number of these stem cells into U-bottom shaped plates and let them self-assemble into spheres. It's not spears, spheres. Then we guide their differentiation in culture and their maturation into full-grown organoids. And in between, we sometimes give them some extracellular matrix for support and better visualization of the neurons. And then we study these organoids at these different stages of the culture, since depending on the length of the culture, they present different properties and different cell compositions and so on. And we use them to study the disease in a patient-specific manner in vitro. And let me show you how these organoids look inside. So I said that they resemble the substantia nigra, and these markers that you have here that are labeled as midbrain identity are the ones that represent the identity of the cells that are specific to this area of the brain. But you can also see that the organoids have other cell populations inside, such as presence of glial cells, which are labeled here by these astrocytic markers, for example, in the lower left panel, or oligodendrocytes in the lower right panel. And you can also see in the plot here in the left-hand side that with the time in culture, the cell complexity of the organoids increases, with more cell clusters appearing in this plot, for example, at 70 days as opposed to when they are about a month old. And they also contain other types of neurons, not only dopaminergic neurons, as in the normal brain, which are labeled here in the panels in the right by different markers for different neurotransmitters. These organoids do not only show the cells present in the midbrain, but also present functional characteristics like synapse formation and also dopamine production, which is the neurotransmitter that is released by dopaminergic neurons. And they also show synaptic activity through spontaneous firing of the neurons, and also neural melanin formation, which is the dark pigment I mentioned you before that is characteristic of these dopaminergic neurons in the substantia nigra. So to further confirm that these midbrain organoids that I'm telling you about actually resemble the area that they are supposed to, we can compare their identity to the identity of an actual human midbrain structure. And this, as you can see in these graphs, the cell populations highlighted here by different colors present in the midbrain organoids are the same populations that are present in the human midbrain. So this shows that the midbrain organoids can be used as in vitro models to study diseases affecting the midbrain. And what's more, these organoids resemble the developing brain, correlating closely with developmental weeks 9 or 10 in the embryo. And therefore, they are a great tool to study early events related to Parkinson's disease. And in the following slides, I will show you this. So, as I mentioned, one of the characteristics of Parkinson's disease is the loss of dopaminergic neurons. So, in the images on your left, you can see that we generated from a Parkinson's disease patient. In the case of the neurons that were generated from a Parkinson's disease patient, there are less dopaminergic neurons than in the healthy control. And in the graphs, you can see that sometimes we are not able to see the loss when we are only able actually to see the loss of these neurons when we create these patient-specific organoids compared to when we just from the stem cells just derive neurons in a two-dimensional manner in a normal cell culture dish. Meaning that the organoids as more complex tissue structures help us to recapitulate better the disease phenotypes. And another key characteristic of Parkinson's disease is, and also common in other neurodegenerative diseases actually, is the presence of protein accumulation in cells. And in the case of Parkinson's disease, this protein accumulation is composed to a high extent by a protein called alpha-synuclein. And what we can see in the plots here in this slide is that the organoids, in the midbrain organoids, we can observe an increase over time of this protein, both inside the cells, but also released into the extracellular space, as highlighted here by the higher intensity of the bands in the first plot and the darker dots in the membrane below. But one limitation of the midbrain organoids as such is that they lack other compartments which are present in the brain, but that cannot be differentiated together within the organoids as such because, as I mentioned before, we use pre-committed cells to generate these organoids, and these missing cell types are arising from other gem layers in the embryo. So I'm referring here to other compartments that are present in the normal brain, such as the blood vessels, and also immune cells, such as the microglia. So having organoids containing these cell types will help us study the key features on the disease as well, since the immune system, for example, is very involved in the pathophysiology of the disease. Therefore, we are working on making these models even more complex by generating what we call assembloids. And assembloids can be referred to as the addition of other cell types, as I mentioned, of different germ layers into an organoid, or as a fusion of two region-specific organoids, such as a midbrain organoid, for example, or a vascular organoid, or a midbrain organoid and a striatum organoid. These assembloids are another tool to study the complex interplay between different regions within the brain, but also between different organs on a dish. So we worked on the establishment of a protocol to incorporate, for example, microglia, which are the immune cells of the brain, into our midbrain organoids by generating independently macrophage precursors from also induced pluripotent stem cells and incorporating them later on into the midbrain organoids. And you can see these in the images by the marker IBA1, which is the microglia marker of these cells in green incorporating into the midbrain organoid. One of the main traits of microglial cells is their ability to take up cell debris, for example, or proteins from the extracellular space to then degrade them inside themselves. So what you can see here in the images and also in the video, if I can activate it, yes, is precisely an example of microglia eating up inactivated yeast particles that are fluorescently labeled for us to be able to track them. And this ability that microglial cells have can be very helpful in the context of neurodegenerative disorders. In fact, we find that, for example, when we look into microglial cells of some Parkinson disease cases, such as patients with the LRRK2 mutation, these cells present an increase of this activity of phagocytosis, and which is shown here in the graph by the increase of a marker which labels phagocytosis. However, this increase in phagocytic capacity can turn out to also be detrimental in the context of disease. And that is by triggering the release of, for example, pro-inflammatory cytokines, which are molecules that will increase neuroinflammatory responses. And we can see this response in our assembloids, in our midbrain organoids containing microglia, where we can see that we can detect these molecules and actually even an increase depending on the context of these pro-inflammatory cytokines. So with all this, until now, I have showed you that the midbrain organoids and assembloids are a valid tool for studying Parkinson's disease because they recapitulate key features of the disease. But let me show you a few examples of how we can use these models and what we can see with them to test the effect of treatments. For example, here we can see the case of a Parkinson's disease patient with a very specific rare mutation in a gene called DJ1, and from which midbrain organoids were derived and treated with 2 different drugs. And you can see in the graph that upon 25 days of treatment, the dopaminergic neuronal loss, which was observed in the organoids without treatment, could be rescued. Another example is a treatment performed in midbrain organoids aimed to decrease the levels of this protein that typically aggregates, alpha-synuclein, with a technique that is called antisense oligonucleotides. And that was a collaboration with AbbVie. And as you can see in the images, the treatment greatly ameliorated the alpha-synuclein burden in the treated samples. And like that, there are numerous publications which support the applicability of patient-specific midbrain organoids for both in vitro modeling of PD, but also for drug testing and development. And these are all the different, for example, assays and techniques that we are able to apply to the midbrain organoids and assembloids to study neurodegenerative disorders, mostly Parkinson's disease, but not exclusively. And all in all, I hope that I could convey the following take-home messages about what are brain organoids as a concept of 3-dimensional cellular structures that mimic the early brain, that we can make them patient-specific thanks to the availability of stem cells. And I hope I could convince you that specifically midbrain organoids recapitulate in vitro the main features of Parkinson's disease. And I also showed you that we can use them to test and develop compounds against different forms of the disease in an effort to make the treatment as personalized as possible. And with all this, I would just like to introduce you the team at OrganoTherapeutics, which is also growing by the years. And I would also like to, no, my slides don't go any further. To thank the organization of the meeting and my funding. I leave also here a couple of links in case you want to check us out via LinkedIn or our website. And I thank you all for your attention.